All good if I share my screen now?
Yes, sir.
Okay, our speaker tonight is Dr. Thomas Cullen. He received bachelor's and master's degrees from the Department of Earth Sciences at Carleton University in Ottawa, Ontario. He received a PhD from the Department of Ecology and Evolutionary Biology at the University of Toronto. Thomas is a quantitative paleobiologist and ecologist. His primary focus is the terrestrial ecosystems of North America during the Middle to Late Cretaceous. And he is currently, within the last few days, an assistant professor of paleobiology at Auburn University in Alabama. So congratulations on that. And the title of his talk is New Evidence for Large Ornithomimosaurs in the Southeastern US and the Biogeography of Cretaceous Ostrich Mimic Dinosaurs. Thomas.
Oh, thanks for the introduction and thanks for having me here tonight. As Dave said, I've just started a position at Auburn. Prior to that, I was finishing up a postdoc at Carleton, and a little while before that, I was actually around at your parts because I was at the Field Museum working on Sioux, the T-Rex, the new exhibit back in 2018, 2019, and doing growth research on Sioux and other theropods then. And I mentioned that because a little bit later in the talk here, when talking about these new ornithomimosaur specimens, we actually apply some of the same methods, histology, and trying to figure out the growth of them. So I will get started now. And before getting into the research itself, I'm going to quickly mention some of the collaborators in this project, particularly Chin Sorig. He was the lead on the project. He published our paper on this a few months back. And then this was a project we worked on together while we were doing some postdoc work with Lindsay Zano in North Carolina. And then the specimens were originally collected by George Phillips and a number of colleagues in Mississippi with the Natural History Museum there. So when looking at the Earth in Late Cretaceous, in this case, about 85 million years ago, as I'm sure many of you are aware, it was much warmer than it is today. It was a greenhouse climate, so higher atmospheric carbon dioxide, warmer temperatures, much higher sea levels, and that relates to some of the warmer temperatures as well as the fact that the way the land masses were at that point and average topography, it was actually easier for some of the ocean water to inundate continental basins. And then because of that, we get this restricted and fragmentary landmass sort of being seen on a lot of continents throughout the world. And if we want to understand the evolution or ecology or other aspects of the biology of different groups of organisms during times like this, and just in general, one of the most important things we have to understand is where they were at a given time, how they're moving around across the world, through what their biogeography is like, what are the forces sort of driving parts of those changes, and how do those change over time. Unfortunately, again, as I'm sure you're intimately aware, the fossil record is unfortunately incomplete. And so we often have gaps, and these can be caused by issues happening before a fossil is even formed. So if material is just in the wrong environment to be preserved, or if something happens to it after the organism dies, it's just destroyed or degraded in some way, so it can't be preserved. Or even after, if it has been preserved, if the area gets eroded away or whatnot, we might never find it, or there might be diagenetic alteration that damages the material such that we can't identify it anymore. In any case, we have to work in these gaps, and the fossil record of many groups of organisms have pretty substantial gaps. Ornithomimosaurs, or ostrich mimic dinosaurs, are one such example. Despite being pretty common in some time periods in some locations, the overall fossil record of ornithomimosaurs is actually very poor. I remember talking to a colleague of mine, Victoria Arbour, when she was talking about how she was frustrated at times by ankylosaur phylogeny because of a paucity of good material at times. And then we chatted a bit about some of the issues I was having with some ornithomimid phylogeny work. And she then said she had a newfound appreciation for her crappy ankylosaur record. Anyway, ornithomimosaurs have a rat-type-like body plan, so similar to large flightless birds today, hence their nickname as ostrich mimic dinosaurs. We have pretty ample evidence now that they were feathered from a number of different specimens. They're sort of, one of their distinctive features is that, again, similar to birds, these guys are often toothless and have a keratinized beak. And it's also been suggested that they were probably herbivorous or at least omnivorous, moving in sort of large flocks or herds, using their speed to avoid predation. And their early record is like similar to many different dinosaurs. They're often very small, known from a number of different locations. In this case, we have fragmentary remains from Europe, Asia, and Africa of a number of different species, all of which had very small, almost peg-like teeth. And then over time, in sort of subsequent ornithomimosaurs that we see appearing later in time, both the sort of derived ornithomimids that are often small and gracile, or the large dinosaurs. In all those cases, we see them lose their teeth. The exact time at which that happens in their evolution is still not totally figured out again, because there's a bit of a gap in their record. But we know that they did have teeth in much of their earlier forms spread across a number of different continents. And then we know that later on, there are no toothed forms anymore. I'll mention just here the dinosaurs, because they're sort of a sister group to the much more well-known ornithomimids. And these guys are sort of oddballs compared to what you'd call, like, I guess, a standard ornithomimid, in that they're often very large, with dinosaurs, the namesake of the family, being the largest ornithomimosaur known and rivaling large tyrannosaurs in terms of size. Again, also toothless. And in terms of the overall skeletal construction, they're much more robust than some of the other ornithomimosaurs. When you get to the late Cretaceous, the one time period where ornithomimids and other ornithomimosaurs are pretty well known, you tend to have a pretty rich record, where they're found with a number of different species known from Western North America and East Asia primarily. And here are a few examples of some of the more well-known species, and a few of the conspecifics of some of those well-known species. So things like Ornithomimus, Struthiomimus, Gallimimus. And they all, again, have a pretty similar design, sort of like an ostrich. But what's interesting is we don't really know what's going on for ornithomimosaurs in general in a number of places, and in particular here in Eastern North America, in this sort of subcontinental landmass that's often referred to as Appalachia, in contrast to Laramidia, the Western landmass, where most of the good ornithomimid specimens are known from. And what's interesting is at various points, Appalachia is isolated from both Europe and Western North America, and at other times it's connected. So trying to figure out what ornithomimosaurs existed in Appalachia and when they had opportunities to sort of move and interchange back and forth with other land masses is a very open question right now. Some of the only evidence prior to this material I'm going to describe as the focus of the talk, some of the only other material known from Appalachia of ornithomimosaurs comes from the early Cretaceous, so about 120 million years ago, and it's pretty fragmentary. It's a couple or a small handful of very fragmentary toe bones, so pedal unguals, little claws at the end, and then some really rough looking rest of the toes. Some of them can be confidently identified as ornithomimosaurs, but not much more than that. They're from Maryland. And then what we generally have is that kind of point record of them in the early Cretaceous. And then we know by the late Cretaceous, about 75 million years ago, that there's a huge diversity of them in the West and in Asia. But then we have this sort of gap in these questions of, for example, what species or groups are present in the intermediary time period. Were they primarily small-bodied? Were they large-bodied? Were they kind of a mix living together? And how do they get around? How are they dispersing? And if we find material in Appalachia, what is it most similar to? So the specific area I'll talk about here is the late Cretaceous of southeastern Appalachia, and specifically the Santonian stage, so about 85 million years ago. As I said, we had a gap in the ornithomimosaur record up to this point, but now we actually have a bit of data to work with. This new site comes from a number of little small pockets of outcrop along the Luxapalala Creek. I always have trouble with that one, near Columbus, Mississippi. And it's part of this broader wedge of upper Cretaceous sediments that crop out across some of the Gulf states. So in this case, we're looking at material from eastern Mississippi, but you get the same rocks cropping out throughout Alabama and a little bit into Georgia. And they broadly represent that sort of paleo coastline, sort of where the Western Interior Seaway is coming down and meeting the Gulf of Mexico. And most of the rocks here are sort of marine silts and muds and sands with a little bit of terrestrial mixed in, often as lag deposits. And in this case, that's exactly what this is. These specimens come from the lower part of the Eutaw Formation, so Eutaw, E-U-T-A-W, not Utah like the state. And again, we're preserving terrestrial materials mixed in with marine sediments along the paleo coastline to shallow marine setting. So this material was transported here, but from the sedimentology, it looks like it wasn't traveling all that far. What work has been done on the site in the past is relatively limited, but there has been some work on the other non-dinosaur vertebrates from here, which is primarily elasmobranchs, things like sharks and rays. And if we look at the primary fossil horizon at one of these sites and look at what fossils come from it, it's a pretty diverse assemblage of marine vertebrates, again, almost exclusively elasmobranchs, and good representation of some of the major Cretaceous shark groups, so things like hybodonts, for example, all being found here. And the dinosaur material was found a little more recently, and this is not all of it on screen right now, but this is the extent of it. So it is still a bit fragmentary, but it represents the best ornithomimosaur material known in Eastern North America at this point, I think, depending on where your cutoff is for Appalachia fossil deposits. And there's a number of characters to identify this material as ornithomimosaur, thankfully, with the toe bones actually being among the most identifiable. Thankfully, unlike many other theropod groups, ornithomimid toes are fairly distinctive and they preserve pretty easily. So one of the things that's, one of the sets of characters we can use for the toe bones here are them being fairly straight and elongate, pretty weakly arched, more weakly developed collateral ligament pits on the sides there, and then having a much less tapered end towards the distal end compared to something like a dromaeosaur or troodontid or in those cases, and then straighter and less curved than something like you would see in some tyrannosaurs or caningnathids or allosaurids. The manual claw at the bottom, we only have one of those, whereas we have a good chunk of different isolated toe bones of different sizes. Manual claw is ornithomimosaur as well, based on its being fairly slender, mediolaterally pinched. Again, a bit straighter, less grooved, and it has a pretty weak flexor tubercle near the proximal end that's also pretty weakly developed, in contrast to a lot of other possible theropods that could be lurking around at this time. The most interesting bone here, probably, even though it doesn't look like much, is this very large second metatarsal, so sort of upper foot bone, that's found with all the ornithomimosaur material at this site. And at first, we thought it might be Tyrannosauroid just because of the size. Once we started looking at it, we were finding ornithomimid characters on it, and we were finding characters that differentiated it from other large theropods that should be around at this time, whether it's large allosaurids, the type of stuff that would have been around prior to the mid-Cretaceous terrestrial turnover, or Tyrannosauroids, the type of stuff that we know is living in Appalachia in the late Cretaceous. And specifically, our main worry was that this was some sort of Appalachosaurus type Tyrannosauroid. But in this case, it's differentiated from it from a number of general Tyrannosaurid characters listed sort of at the top there, but in particular from Appalachosaurus by how elongated this midshaft of the metatarsal is, proportionally much more so than in Appalachosaurus. And that is, again, much more consistent with what we see in other ornithomimosaurs, including some of the really big ornithomimosaurs, things like Beishan Long. Now, I mentioned like some of the smaller ones or larger ones. I'll just start going through here and giving you an idea of how large some of these things are, as well as discussing how there's actually a bit of a range of sizes. So we're not just finding isolated, or we're not just finding assorted bones that were probably from the same individual, or finding bones from at least 2 to 3 individuals based on the different sizes that we're finding. And one of the first questions is, because the material is pretty scrappy, are the available bones here sufficient to get a good idea of actually how large in body size these things were, even though we only have like toe bones or broken leg bones to work with? So the answer to that question is mixed. Ideally, to get something like a body mass in a theropod, what you'd want to have, like, well, a complete skeleton would be great, but if you have to work with isolated bones, something like a femur would be ideal, because there's a very tight relationship in terrestrial vertebrates between the circumference of the femur and the mass of the organism. That's because of the way the femur is acting as a weight-bearing bone and growing fairly symmetrically. And because we don't really have femora here to work with, what we needed to do was to use linear regressions to try and estimate and predict what the femur circumference of each individual that we're identifying from the site is based on the proportional relationship between femur circumference and the length of bones we do have, using examples of more complete specimens of various different ornithomimosaurs, where we can, establish that relationship and then plug in our samples where we have only one of the two variables and then predict what the other one is. So that's what we need to do. From the linear regressions we're doing, we can then, as I said, estimate a missing value. One of the caveats, there's sort of two main caveats for this. First is, as you can see from the sort of diagrams there and the headings at the top of them, we did this both with and without Dinochirus. So Dinochirus is so much larger than any other ornithomimosaur that it has a very outsized impact on the results of these regressions. If you include Dinochirus, it tends to hold the estimates really larger because it's such an outlier. Whereas if you don't include them, then you get smaller values, but you don't know if that also is itself an artifact because you're excluding the potential that this thing could be larger than any of the pool that you're looking at. So we did it both ways in order to get an idea of the range of possible sizes here. And the last problem is that some of the elements we're using to do this based on necessity, because it's all we have, things like toe bones or metatarsals, they are under different allometric pressures. Different allometric factors are affecting their growth compared to something like a femur. And so using those to directly estimate a femur circumference is not a guarantee. And I'm saying all these caveats up front because some of the numbers are going to be here very large. And I don't want you to think that this thing was like Dinochirus size, because it almost certainly was not. But the point of doing this is to give us a rough estimate of what the range of possible sizes of these Mississippi ornithomimosaurs are. I'm also saying this because when the paper came out, there was a bunch of people talking about how this thing's like giraffe size, and it absolutely is not. That was just like if you take the biggest number and go way outside with it. So if we estimate possible femur circumferences from these and then translate them into masses and break those down again, because Dinochirus is so different, into estimates with and estimates without Dinochirus, we get numbers like this. So we primarily have two size morphs. We have multiple individuals here, but we have sort of two rough size morphs. We have the small material that's kind of roughly the size of a lot of the small ornithomimus that you find in places like Montana and Alberta. And then you have this large morph, which is primarily, but not exclusively, represented by that large metatarsal, also some toe bone, like pedophalanges. And if you include Dinochirus in the estimations, then you get really large mass values for the large taxon. So for up in the area of 9,000 or 800 to 9,000 kilograms, which is obviously a really big range. And the pedophalanx, as I said before, it's the least reliable. So again, hope I'm not like being too much of a broken record, but like don't treat this as like gospel truth sort of mass values. The important thing though is in the small morph, the estimates are probably pretty reliable because it's falling sort of in the cloud of the other data. So the regression there is going to be a little bit more reliable because we have data on both sides of it to kind of hem it in. The large one, again, in case is sort of on the far end, so it's more prone to being on the one end or the other of the confidence interval. But in any event, based on the metatarsal and tibia values that we are estimating from, we get something like 140 to 180 kilograms for the smaller morph, something like 300, 800, maybe 1,000 kilograms for the large one. Potentially a little more than this if we really go off further, but the 9,000 value is almost certainly an outlier. But this sort of range of mass differences means that we have one ornithomimosaur here, individual, that is about the size of things like Ornithomimus, and we have one individual, at least, possibly two, that are much larger and more in the Bayesian long sort of range, so almost double the size. And the question there then becomes, if we look at the size classes represented compared to other known ornithomimosaurs, so here, the small one, versus here, the large one, where it's, again, the size of something like Bayesian long, or the very largest adult specimen, Gallimimus, the question becomes, are these an ontogenetic series? So is the small one a juvenile of the large one? Were they part of the same sort of herd, for example, the same species, and they kind of got washed in here together? Or does it represent two different species? Are there at least two different species of ornithomimosaurs present in Mississippi at this time? Well, the way to get to trying to solve this, or at least one of the ways, aside from finding, again, more specimens, is to use bone histology. And the reason for this is that we can use the microstructure in bones and the patterns of change in that microstructure to estimate things like the rough age of the animal at the time it died, its relative maturity, and some of the growth rates of that animal. And you can do this with living animals, extinct animals. And it primarily works because, somewhat analogous to trees, many fossil vertebrates will have annual growth slowdowns or cessations that leave a visible mark in their bones, sort of like a tree ring. And we can use those as our annual markers to get an idea of how the growth is going in the preserved interval of the bone. It's not perfect because, unfortunately, unlike trees, as the vertebrate or amniote bone is growing, that middle marrow cavity is also expanding. And so some of the earliest records of an animal's growth will often get sort of eroded away by the expanding marrow cavity and prevent that from being preserved, which is why having ones at different ages can be helpful. But what we would predict here is if these things were two different specimens of the same species, you would probably have the smaller one having considerably fewer growth marks than the larger one. And with the rates of growth that are evident from them being such that you could reasonably go from the small one to the large one without having to have any really crazy growth changes. And alternatively, if the growth mark counts are fairly similar, despite them being very different in size, to the extent that it's extremely unlikely that the small one could grow into the large one in the number of years available, then it would suggest that they're probably two different species. But before getting into that, I'll just briefly go over a bit more bone histology, so we're all on the same page with how we do this and what we're looking for, and then loop back to actually interpreting what the cut bones look like of these ornithomimosaurs themselves. So one of the big things we're looking for here is differences in the types of bone tissues. So generally speaking, the differences in bone tissue will reflect differences in the depositional rates of the bone by the cells as the bone's growing. And so if you have really, really highly organized bone that's growing very, very slowly, the cells are being very carefully putting everything down, then you get what's called a lamellar matrix, like the bottom left. If you're growing very, very fast, it's very disorganized, then you get woven fibered bone, like the middle image. And then you can also have circumstances where you have what's called parallel fibered bone, which is a bit of a mix, an intermediate between the two. And then you can also actually have two of these types, so the lamellar and the woven fibered, largely being combined with like a basic lattice that's then being filled in more slowly afterwards with what's called fibrolamellar bone. That I mentioned fibrolamellar bone because this is the type of bone that you see in a lot of large animals that have phases of rapid growth. So things like birds and mammals today, and things like dinosaurs in the Mesozoic. And when you're looking at fibrolamellar bone, depending on just how fast that bone is growing and how disorganized it is, you get these different patterns of vascularity. So where the different blood vessels are and how organized they are in that bone structure. It's the same sort of idea. Generally speaking, really poorly vascularized bone will be growing a bit more slowly, not always, but often, and it would be reflective of physiological differences. And so when you have things like longitudinal bone, where you just see a series of little holes with no interconnecting network between them, that just suggests the vascular pattern is fairly simplistic, and the bones are just sort of, or the vascular canals rather, just sort of tubes underneath that surface running along the long axis of the bone. And in the more vascularized examples, so say something like plexiform or reticular, you have those initial channels, but now they're anastomosed and interconnected between each other in increasingly chaotic ways. And so again, we can look at that and use that in conjunction with the counts of growth marks to get an idea of sort of how fast these things are growing. One of the other differences is the idea of primary versus secondary osteons and blood vessels. And so when we're trying to look for things like growth marks, we're looking in the primary bone. This is looking through our primary osteons when the bone's initially growing. When we get secondary osteons, these will look like a little point with a bunch of concentric rings around it. So something, I don't know, can you see my mouse, hopefully? I'll just annotate. Yeah, draw, here we go. Something like this. That's a secondary osteon. So I have that little center point and then lots of these like concentric rings around it. That's useful and indicative because it's telling us, how do I get rid of this? Okay. It's telling us how mature the animal is. If you have extensive secondary remodeling, like in this example from Sue the T-Rex at the Field Museum, then you have an idea that the animal is very old, very mature. If it has multiple generations of these secondary osteons sort of cross-cutting on top of one another and completely obliterating most of the original primary bone, that can tell you that the animal is quite old and probably not growing anymore. The downside of that is that if you do have secondary remodeling in your bone to the extent of something like Sue, that means that in some of the bones, you'll simply not have any real growth record, period. And so it's a double-edged sword, which is why in the case when we were studying Sue's growth, when we used a bone like this, the fibula, it could just tell us that it is mature and not growing and nothing more than that about the age, but something like the rib or the femur were able to tell us how old it actually was because it remodels at a different rate and it was still quite remodeled in the center, but the outer part had enough growth marks that we could do something with. Now, yeah, as I said, the presence of remodeling can correspond with the loss of the growth record. And this is a problem when we're trying to do skeletochronology, which is our main goal here for aging these organisms. And so the main thing with skeletochronology is we're trying to look at those growth marks, the lines of arrested growth or annuli, and the zones between them, which is the actual amount that the animal has grown in a year. And then we also consider something that's called an outer circumferential layer or an external fundamental system, which is sort of like a rind on the outer part of the bone. And that is another indicator that the animal has stopped growing. If you reach your sort of end of your growth pattern and you're really slowing down, your body will still be putting down these lines every year, but there'll be basically no growth between them. So you just get a very tightly packed, almost completely avascular zone at the outer margin of the circumference of the bone that just has like tiny little line after line after line after line that tells you that it's not growing anymore. If we see that, we can be very confident the animal is done growing. Otherwise, it's a little harder to tell. And the last thing is that by tracking some of these relative changes in the spacing and circumferences of the bone as it's growing, that can be used to infer aspects of relative growth and osteogenetic stage. And this is what we often have to do because we almost never have a complete growth record of an animal in a single section. So if you have something like on the left, constant spacing where the radius is exactly the same every time, that means that the animal is actively growing and that the growth rate has to increase year after year after year. So it's in the fastest part of its growth phase in order to keep a constant radius despite an expanding circumference. In the middle example, if you have very small, steady decreases in spacing, that suggests that the growth rate is remaining relatively constant. And as a result, with increasing circumference, the radius is getting a tiny bit smaller each time. And then the example on the side or on the right, where you have really wide spaces that are suddenly decreasing down to much more narrow spaces without any kind of steady gradient, that suggests that your growth rate is decreasing. And that is more reflective of what you see in a lot of different amniotes and mammals and birds and dinosaurs, that they'll have a very rapid growth spurt sort of period, generally relatively early in their life, up to sort of teen, like subadult sort of stage, before petering off very quickly and reaching their adult size. So back to the Eutaw ornithomimosaurs, we primarily sampled two bones, that really large metatarsal, which we cut twice, once at the midshaft of the bone, and again at a position near the proximal end, which we did because the midshaft of this specimen unfortunately has a pathology on it. So we wanted to take two samples to get another look at the same specimen. Oh, hold on one second. My cat's making noise here. Here, get in. Come on, buddy. Sorry for the interruption. With the other specimen, which is one of the small ones, we were lucky enough to actually have a tibia, which is usually a very good bone for sectioning. We cut that at the midshaft. So here's what the histological data look like. The top two examples there are the same bone. They're the big metatarsal from the largest specimen, with the cut at the main place you normally cut in the midsection, as well as higher up at that proximal area. And then the last one is the small specimen. So starting with the proximal section of the largest specimen, it has about 5 growth marks preserved. Sort of see them. Some are more distinct than others. There's a bit of a crack happening right along this one. Some of these others are a little harder. If you go down to the mid-cut section of that same large metatarsal, the legs are a bit easier to see. So you have one there, a couple others. There's a zone of some pathological bone, which I will explain in a moment. And then you get up to these sort of outer ones. And the outermost 4 or 5 are pretty easy to see. You can see this dark banding. Those are your annual growth marks. And so we see 9 total in this specimen. And again, to do a little brief diversion here, I'm going to talk briefly about the pathology that we see on this. I don't know if you can see it there, but you have the actual original cortex of the bone, and then you have this massive semicircular bulge of pathological bone on top of it. And this is a CT scan of the specimen to show you what that looks like. The blue is where we've digitally reconstructed the original bone, or rather just sort of remove the pathological bone, with the green pathology on top. And this is showing the inside of what it looks like. This is a pretty gnarly pathology. I'm going to focus your attention here, these 3 spots. So right here, the bottom left, you can actually see a space here where the original bone was broken, and there's infilled with this pathological, very porous material, porous bone in there. And the original top of this metatarsal is sort of floated away from the rest, and all around it has filled with this really gross, pathological, fast-growing reactive bone tissue. There's also another spot on the same bone where you have a bone fistula, where these 2 areas here, the inner cavity of the bone and sort of the outer space are connected now. And you have a bunch of osteonecrosis and dead bone around that as well. What my colleagues who were working on this, as we were sort of doing these projects in parallel, what they were thinking is that this represents a pretty severe blunt force trauma to the foot of this animal and its metatarsal, that caused it to break and then heal pretty poorly and develop this big reactive bone callus on top. The good news is, because of the way that the injury appears to have happened and the way that the reactive bone has grown on top of it, it doesn't actually seem to have impacted any of the preserved growth mark counts. In such a case of an injury like this, if a lot of the original bone ends up getting destroyed or resorbed or really badly damaged, it's possible that it could remove a good chunk of your original growth record through rapid secondary bone growth. But in this case, it didn't. It just sort of displaced a big piece of the bone and then other stuff grew around it. So that's good. Really awful injury. I'm sure this animal was in a ton of pain, but it survived it at least for a while. And we know that it lived at least 9 years. And because we're missing a bit of growth in the middle, presumably more than that. So back to the skeletal chronology question, we compare the 9 growth marks that are preserved here. And now we look at this small specimen. It has 6 pretty unambiguous growth marks. It doesn't have any pathologies. It's just a pretty nice section and pretty sort of standard ornithomimosaur looking bone section. It actually looks a lot like a ornithomimosaur bone section of a tibia that I made myself in another study from 10 years ago or so now from Alberta. I guess it makes sense. They're both ornithomimosaurs, they're both tibiae, but it looks almost exactly like that section, except having about 3 times as many legs. And so again, the summary here is somewhere between 5 and 9 growth marks in the larger specimen, 6 in the smaller one. And to give you an idea of what the size difference is here, again, the center and left are the same individual, just different places on the bone. And then on the right is the tibia of this smaller individual. And again, usually a tibia, circumference-wise, is going to be quite a bit larger than the metatarsal of the same animal. So that to further underscore, this thing is quite a bit smaller than the large morph. And yet, despite this, and despite them both seeming to grow pretty rapidly, they have relatively similar number of growth marks. The larger individual has a few more growth marks, at least at the mid-cut or midsection, but it's similar enough that it's very unlikely that it'd be able to reach a size like this in a number of missing years without a really massive change in its growth rates to the extent that we have no record of in any other sort of theropod dinosaur group. So with that, you can probably say that it's most likely that they represent two different co-occurring species rather than a juvenile and an adult. And this is, again, it's not entirely unexpected, but because we have other formations where this has been observed. You think of like Dinosaur Provincial Park in the Dinosaur Park Formation in Alberta. We have 3 to 4 species that co-occur for at least a little bit of time. In the Nemet, even more substantially in Mongolia, you get things like Gallimimus and Ceramimus and Dinocyrus all co-occurring with different sizes. So ornithomimosaurs can co-occur in different times and have been recorded doing so before. But if at this point we had no record of that anywhere in Appalachia, and really no record of any kind of what the diversity of these organisms were there. So the next sort of question, which we were hoping to be able to answer with this, was where do these things come from? We can get a little bit from the time periods we're looking at and what the geography was like on Earth at the time. So the two main options we have are that, or did they come from the West through Laramidia and then eventually back from Asia? Or did they come from somewhere else in Eastern North America or somewhere like Europe and elsewhere around there? For the first scenario, coming from the West, on its surface, this seems like the more probable because we know that there's a record of ornithomimosaurs throughout Western North America. The only issue is that we have that very large gap in the record of ornithomimosaurs in Western North America. There's a couple of kind of, not dubious exactly, but debated ornithomimosaur taxa from much older units in Western North America. And then you don't really get really unambiguous ornithomimids in Western North America until after the Santonian, until the early Campanian, after these things had already existed in Mississippi, and importantly, well after total separation of the two subcontinents by the seaway. So it's not impossible they were coming this way, but it appears right now that it's more likely that these things were dispersing either from somewhere else in North America to get to down here, and then originally either coming back from the Western North America way earlier, or more recently, but from somewhere like Europe and sort of in east towards Asia. But the problem with this is that we can't really go much further than this at this point without more data from places like Europe, where unfortunately the record is really patchy because, as you can see at the time, a lot of it was underwater. It was sort of patchy, occasionally connected chunks of land, where we know from other groups of organisms, various ornithischians, that animals were dispersing through all of this network and making it across these continents, but we aren't able to say for sure yet with the ornithomimosaurs if that's what was happening. So just to summarize, the Eutaw Formation ornithomimosaurs here are pretty just important because they provide us with our first record of Santonian ornithomimosaurs in North America. And if you look on this diagram here, this is showing all of the known ornithomimosaur taxa that up to date, with their vertical position representing when in time they are in the early and late Cretaceous, and then their horizontal position being what continent they're from. As you can see in North America, there's a missing gap in time from about 115 or so million years to previous to this until about 75 million years. And now with the Eutaw ornithomimosaurs, these two groups, we have this gap in the Santonian sort of now filled with something. And up to this point, there hadn't really been any ornithomimosaurs anywhere known from the Santonian because there's a gap at the same time in Asia as well. Again, in terms of size classes, our histology suggests two coexisting species one that was the size of a very large Gallimimus to a Bashan long sort of size. So that's this one here in gray. And where's Gallimimus on here? This guy over here, also in gray, they're about the same size. So two of the larger ornithomimosaurs, but not the largest. And this thing was definitely not getting into Dinochirus sized, but still larger than most ornithomimosaurs were. And then the smaller one was much more of your typical ornithomimosaur in size, being similar to something like Ornithomimosaurus ratavates. And what they were doing in terms of ecological niche partitioning, the size difference probably allowed them to access different plant resources and other resources that they could probably avoid competition in that way if we go by the co-occurring ornithomimosaurs in other areas as a parallel example. And again, the biogeographical dispersal patterns still remain a bit unresolved, and we're hoping that with additional sampling, we'll be able to get into some of that. But for now, I would say that the dispersal option coming from Europe or other areas from the East appears more likely than coming from the West, given the seaway separation and the differences in the sort of morphology and size of some of the taxa that we're seeing here, and their similarity more to some of the earlier diverging groups from Europe and Asia. And so with that, I just want to acknowledge our colleagues and people who helped us, and thank all of you for listening. I can take care of some questions now if you'd like.
Do you pay off fast by ostriches too?
Pardon me?
Do you pay off fast by ostriches, but they think they are? Because they're bone mimic, they look like ostriches, do they run fast?
Oh yeah, they probably run very fast. The construction of their ankles and their lower half of their legs suggests they were probably really good at running.
Yeah, when you saw the part of the bone with the infection, my mind told me Carl thought it could be disease could kill that dinosaurs. Because it's broken, probably disease killed it.
Yeah, it's very possible. We don't actually know, but that is definitely possible.
What evidence do you see of feathers?
In these specific specimens, we don't, Irene. And also, hello, nice to hear from you.
Hi, Tom.
But there are a number of specimens of ornithomimosaurs from, there's 3 or 4 now in Alberta, in the Dinosaur Park Formation, that have preserved feather impressions on them. And then I think there's one in Mongolia now as well. I don't know if it's published, but there's enough specimens now that that have them. We're reasonably certain that the whole group did, considering sort of where they sit in the phylogeny. But the specific Mississippi ones, we have no direct evidence of feathers on those specimens.
Thank you.
That first cross section of bone that you showed, it looked different than the other ones. Was that from something else? You go back to the very first slide where you showed a bone. Keep going. Keep going. Yeah. Keep going. No, keep going. Keep going.
This one?
Yes.
Yeah. So this is a Tyrannosaur. I just chose it because I like this one because you can see the legs really nicely.
And that's also an injury then too on that one? Or?
No, this one's not actually an injury. This is kind of interesting. This is so this is also a metatarsal. This is a 4th metatarsal, I think, of a small Tyrannosaur. It's at the Field Museum, actually. It's specimen FMH PR2211. It's the one that's called, I think the nickname is Elmer, after Elmer Riggs. It's a small Gorgosaurus. Anyway, all those little like marks on the bottom and on the right of it, the different sort of visual appearance, that is secondary bone growth that has occurred sort of prematurely because of strain, because that's where some of the ligaments attach to that metatarsal. And so that you get this sort of band from the middle to the outer part of the bone being under extra stress, and so getting sort of repaired and regrown more often than the rest of it. You see that in certain bones.
Thank you.
I have a question for this type of bird I'm sorry, dinosaur. I'm confused about the fact that these are herbivores, but at such a large size and yet lacking molars or anything like that to digest. I'm just trying to figure out what would they be eating and how much could they do to reach those larger sizes. without molars to grind and, because we don't see that really, that large size, without the teeth. So I'm just kind of trying to.
Yeah, so it's a good question. I mean, they're using, in order to break up plant matter or whatnot, it's hard to kind of see on these, but you can see a little bit on the one at the top left there of just the isolated head. But they have like a keratinous horny sheath on the end of their mouth, sort of like a bird's bill. And so with that, they could probably cut up leaf material. I mean, in the same way that an ostrich does. Like ostrich today are primarily herbivorous as well. But you're right. I mean, it would probably be a bit more difficult from the process really tough plant matter. So they're probably eating like easier to digest material. They're not eating grass. Grass also is not really a thing at this point. And it's what's been suggested for the really big ones like Dinochirus is that it may have been eating like swampy aquatic plant materials. It's also possible that these guys were omnivorous and eating more than just plants, but an omnivorous diet has been suggested based on not only the beak but also some of them having like fine rock material in their gut, same way that some birds do when they eat like gastric stones. So yeah, I mean, it wouldn't be as efficient as something like a grazing cow or horse today, but if it's enough plant material for them to access, particularly in like aquatic plants or ferns or things like that, they could probably get by.
Is there anything in their foot structure that would support the idea that they're semi-aquatic?
No, and I'm not trying to imply that they're semi-aquatic.
Well, but I mean, if they're eating aquatic, that would imply that they're wandering through an aquatic environment to find. I mean, so that's what I'm trying.
Yeah, but I mean, it would probably be more similar to something like a heron in that, like the feet are splayed out rather than being like webbed or any kind of thing to actually move in deep water. But no, there are no direct data to suggest like a semi-aquatic or really strong water movement. Aside from, I suppose that they, with the exception of Dinochirus, who's I guess unfortunately on screen now, most ornithomimids have very proportionally long legs, which is something you see in animals that move in sort of shallow water. If they're sort of bending down to grab stuff out of the water, like a heron and other sort of wading birds, they will typically have long, skinny legs. And all these guys have very long, skinny legs. But I wouldn't go so far as to say that's necessarily an indicator of aquatic movement or wading or anything like that necessarily, because it's also something that you see in animals that do a lot of running, which
Right, like an ostrich or something.
Yeah. And in terms of another thing for herbivorous large beaked things is we do also have things like moas and elephant birds that went recently extinct, which were not quite Dinochirus size, but easily the size of things like Gallimimus and Bashan-long. So we do have, even in a fairly recent history, evidence of animals without teeth being able to reach pretty large sizes and subsist on what appears to be a mostly plant-based diet.
I remember one episode preached part of the Onoromimimus. They think they behave like ducks and geese because of the beaks.
Yeah, I've heard that suggestion too, that sometimes instead of eating just plants, they might sort of grab down and then sieve small invertebrates out the way a duck or a goose would. That's been suggested with a few species where there's some kind of grooves on the inside of their bill. But yeah, it's certainly possible.
Is it possible they broke a metatarsal on their foot when another dinosaur stepped on their foot, or do you have any idea as to how these could have gotten broken?
We don't know exactly, but it's funny you mentioned that scenario, because that is the one that was considered the most likely when they were looking at the pathology, that something else stepped on it. It was consistent with a stomp wound, if you would see in other animals or in people. What exactly may have stepped on it, they don't really know. Something similar sized, but
It is pretty amazing though that it survived, and
it must have been an absolute agony. Like I'll show you the picture. I'm sure it gives the impression correctly, but it's really gross how big the lump on this thing is. Where is it here? Yeah, like just on the left there, like the original bone, like the thickness is almost the same of reactive bone as it is the original bone. It's really wild. And yeah, so it's amazing it was able to survive at all. And yeah, as the other fellow was saying, it's very possible that it died of this injury eventually, but we don't know.
Would that reactive bone have grown pretty fast? Like, can you
Yeah.
see if that was laid down pretty quickly?
Yeah, it would have grown quite fast, but I don't know exactly what the timeline on it would have been. But it wouldn't grow like in days or anything. This thing would have still had to have been alive for months, maybe year, but it'd probably be growing in a matter of months, is my understanding.
You know how Sue has that big bone kind of disease on its fibula?
Yeah.
Is that probably a similar type injury, do you think?
Yeah, there's been a number of things suggested for what was the cause of the pathological fibula on Sue. I've seen like an injury from a prey animal being thrown around as one of the most likely scenarios, but as far as I'm aware, there's not like a strong amount of evidence to one particular scenario versus the others. But I've seen it suggested that as in a number of Tyrannosaur specimens that have a broken fibula like that it could have run afoul of like an ankylosaur tail club, or it could have been an injury from some sort of intraspecific behavior with another Tyrannosaur, or as the Sue exhibit suggests, it may have been gored by a Triceratops it was trying to eat. We don't really know. I think the injury on Sue's fibula is such that it's not consistent with actually being directly stepped on. It was more of like a lateral hit rather than something coming down on it. But other than that, I don't know if there's any hard evidence to suggest a particular scenario.
I'm amazed. I look at some of the smaller dinosaur bones that I have, which aren't that remarkable at all. And I'm amazed that you're getting so much detail down to how the vascular orientation, and that just boggles my mind.
In the bone microstructure, yeah, no, it's really cool what you can do with some of these histological techniques. The downside with them is that they are unfortunately destructive. The hope is that one day our like CT and synchrotron scanning will get to the point where we don't need to do any cutting at all anymore. We're not quite at that point. Even the best synchrotron data, the like voxel sizes are still too big to make out some of the really fine scale details. But I'm at least hopeful that as we get better with it, we'll be able to at least get to the point where we can identify those growth marks without having to cut things. That would make everyone's lives a lot easier and save me a lot of paperwork having to beg curators to let me cut stuff.
Yeah, it just amazes me that there's so much detail available when I look at them and just go, wow, I can't even imagine seeing what you're seeing. That's
what's pretty neat too is if you find, I find toe bones work pretty well for this because they're very dense, but it works with limb bones too. If you find a one that's broken out in the Badlands or whatever, and the surface of the break is pretty clean and like perpendicular to the length of the bone, you can sometimes see these growth marks with the naked eye because of the textural and kind of color shift differences on that fresh broken surface. But it's not 100%. But like with some specimens, when the staining of the bone is just right, you can make them out pretty well, which is something I really enjoy, like walking around being like, oh look, you can see like 2 or 3 growth marks on it, and then oh, it's a
Did you get to do the histology yourself?
On these particular specimens, I didn't. Chinsorg did. I've done histosectioning on maybe 15 or 20 theropods before. I worked with the slides and did all the interpretations on them. But in this case, when we finally got permissions to cut those 3 specimens for a number of reasons, like pandemic and then a few other things after, I was no longer at the North Carolina Museum, and Chinsorg was, so he did the individual cuts. Him and also Lisa Herzog, who was the sort of head lab manager there.
How big is the cross section prior to being polished and ground?
Big how? Do you mean like?
Is it how thick? Yeah.
It depends. In most of the ones when I do them, it's usually maybe
Eighth inch or
yeah, sorry, I do everything in centimeters. I usually do it like maybe like a third to or half a centimeter.
okay.
In some cases where it's like a really important specimen and we only get sort of one chance to cut it, and we want to have a little bit of wiggle room if like the piece that we take out, we can't get another one, I'll do one that's maybe a centimeter thick if I can get away with it. And then you have like a nice thick puck to make 2 slides at least.
Yeah.
Something we've also been doing recently when we do take a thicker piece out is we have a good idea of the kerf loss on the blade of how much bone is being totally removed. And we will make a mold and cast of the piece that we're removing. And we'll put it back in with enough epoxy to approximate the kerf loss. And that way we restore the dimensions and the morphology of the original bone.
Thank you.
So I have another question. I'm still trying to sort in my head the bone growth and all that. I know that and I'm just going to pull up modern birds. They reach their adult size rapidly.
Yeah.
And when you have mammals, it takes time for many of them, years to reach the adult size. And most of them at that point stop growing. But then you get into some of the reptiles, which, for example, the anaconda, they will keep growing, or the river, the Nile crocodile, they grow after each meal. So their growth spurt is like once a year, or when the animals migrate around them and they've got the feast or famine, that's when their both growth is. So I'm trying to figure out what you're finding out from the study of the bones and the growth as to how these animals are growing.
Yeah.
Does that make sense?
Yep. Yeah. So first off, the thing with the like reptiles kind of growing forever and it being like really dependent on the resources in any 1 month or a year, that's actually a bit of a thing that was long believed and is slowly the data are suggesting that they don't work that way. It's just that they have a what's called determinate growth. So there is sort of a set endpoint the way a bird or a mammal. It's just that it's so far out that a lot of them never reach it in their life because they're growing comparatively slowly. But aside from that, you're absolutely right, though, that because they're having a very feast or famine type lifestyle, their growth gets affected by that. But if you were to plot out like a graph of like what their growth should look like kind of thing, and if you had one that was well fed forever, it would eventually stop growing. I just mentioned that because there was a big study that Hollywood Bird and a few others did some years ago that sort of countered that long-standing idea that like if you had an alligator that was growing for like 100 years with this massive, but there is a point where they do stop. But so dinosaurs like this tend to actually grow a lot more like a mammal than a bird. Birds are pretty unique in that they grow so fast that their bones often don't even preserve a growth mark like you see on the screen here, because they only get that like outer rind with the little tiny stacked marks, but this sort of nice like several marks of differing thicknesses between them. A lot of birds never show that because they grow so fast that they stop growing before a single year's gone by. Ostriches are different, and a few the other larger birds where you do get a pattern a bit more like this. But the animal that I've seen that grows the closest to something like an ornithomimid like this is actually various cows and deer and horses, stuff like that. If you compare the thin sectioned bone of like a femur of a white-tailed deer with a ornithomimid, there's a couple of minor differences, and obviously the shapes are different, but in terms of the bone structure and the pattern, it's almost exactly the same. It's uncanny. And so from that, we're basing some of our ideas of growth rate changes using that as a bit of a model. It's not a one-for-one, but the change in the vascularity patterns, the change in the spacing, in the number of growth marks in most ornithomimids and in a lot of sort of medium-sized theropods like this tends to look a lot like a medium-sized mammal. And so it's probably along the same lines, high, fairly high growth rate, reaching maturity in some like relatively small, but not zero number of years. And then probably also a fairly high metabolic rate.
All right, thank you. Because I've been trying to figure that one out. And thank you.
Yeah, I mean, it gets a bit more complicated, unfortunately, because as I said, that middle area of the bone, the like marrow cavity or medullary cavity, there's information missing in there. So of course, if we use the one that's on screen right now, when this thing's born, the circumference of its metatarsal is probably like an inch around or something. And the hole in the middle now is like 3 or 4 times that easily. So there's certainly a period in the first year at minimum where you're completely eroding the earliest bone, because as it's growing, as new bones grow on the outside, the inside is eating away as the marrow cavity grows.
Have you been able to find enough bones of different ages of the same species to see that?
Yeah, that's actually one of the 2 main ways that people do what's called retrocalculation, which I didn't really talk about in the primary discussion just because we don't have the ability to do it in this case. But if you have enough specimens to overcome the kind of individual variation that you can get, then you can, sort of line up the one of all the different ages and get an idea of what you're missing. The other way to do it is you use a series of statistical models, and you fit a bunch of different like growth curves with different parameters that have been developed from living animals, and you fit those to the data you do have, and it kind of tries to back estimate what's missing. And you can do that with multiple individuals at different stages as well to refine it. But yeah, there's a few different ways to do it. The more traditional ways to do is to just sort of like Russian nesting doll them. But then you run into issues of if there's individual variation in size, as we know exists in all animals, then it's a little harder to actually predict something from it.
Thank you.
May I ask you, what's your cat's name?
His name is Theo.
Theo, oh, I was expecting Tyrannosaurus or something.
No.
Oh, come on, I want to see him.
He's not in the room anymore. I don't know where he is.
Oh, that would be oh, that would be cute to see him. Well, I love cats. They're cute. I would know that.
That's true. I love them too.
Yeah, but I like to take them, but my dad is allergic. So is my sister.
Oh.
I'll get a cat.
At this point, I'm going to stop recording. And if there's any more questions, please continue. Otherwise, we can just make this open for discussion for anything. Thank you.